Let's start with biological activity. But what is it? Biological activity is the effect a given substance or drug imposes on living matter. This impact is related to physicochemical and structural properties of the active substance. Among the various properties of chemical compounds, biological activity plays a key role in medical and biotechnology applications. What in fact is a bioactive compound? A bioactive compound is a compound which has the capability and the ability to interact with one or more components of the living tissue by presenting a wide range of effects. The origin of these substances can be natural, terrestrial or aquatic, or synthetic. And why not? Maybe one day we would have bioactive compounds of extraterrestrial origin. However, bioactive compounds may show some adverse and even toxic effects which may prevent their use in medical practice. The biological activity can be expressed in two ways: by the dose or concentration of the given compound that causes the standard biological response, or by percentage response compared to the control at the standard dose or concentration. The first method is the most commonly used one. The basic measures of biological activity are LC50, IC50, EC50, LD50, ED50, MIC or MTD. Are bioassays. These are procedures for determining the concentration or biological activity of a substance by measuring its effect on an organism or tissue. Bioassays were used to estimate the potency of agents by observing their effects on cell and enzymes in vitro methods or on living organisms in vivo methods. In turn, ex vivo assays refer to experimentation or measurements done in or on tissues from an organism in external environment with minimal alteration of natural conditions. During organism selection, in case of in vivo assays, it is recommended to focus on the so-called 5R rule. This rule features certain criteria that should be met by the perfect test organism. Moreover, the test organism should be easily cultivable in laboratory conditions. Additionally, it's good to possess the knowledge of organism genetic structure and its culturing history. Mostly these criteria also apply to the in vitro methods. Drug discovery process could also be accelerated by applying high-throughput screening. This method is also being set up for academia and some research hospitals. Independent of the applied screening technology, high-throughput screening is based on the same fundamental principle of performance management as time, costs, and quality of the process. As a result of increasing number of chemical compounds and molecular targets for high-throughput screening, there has been a trend towards the miniaturization of assays to enhance assay throughput and screen times. In the early 90s, when HTS methods were introduced, 96 12 plates were the primary format for screening. A decade later, there was a push towards miniaturization. Then, 384 well plates and 1,536 well plates have come to use. Further still, some progress has been made in increasing plate density up to 3,456 wells. Current trends point towards the 384 well and 1,536 well plates as industry standards. Time to sum up. The graph below presents the relationship between throughput and biological relevance of different kinds of assays. Biochemical assays and cell line assays can be characterized by high capacity but by low biological relevance. On the other hand, tests with the use of tissues, animal models, and organoids give a high biological response. However, here the throughput is low. HTS assays with the use of whole organisms would be an optimal solution as they combine both high throughput along with adequate biological relevance. And what is important, they have started to develop very quickly in recent years.